Hello and welcome back. We are moving on to the next part of chapter 7, which covers neuron anatomy and physiology. So we're going to take a look at the structure of the most common type of neuron, classify different neurons based on their function and structure, and then we'll take a look at how neurons fire, these electrical impulses, these action potentials that trigger that electrical communication that runs along the length of the axon. All right, so let's get started. So this first diagram shows you what's called a typical neuron. So this particular neuron is called multipolar, and we're going to talk about why that is here in a little bit. But let's just focus on the structures for right now in this picture. So when we take a look at the neuron, it's a really interesting cell. It doesn't look like your typical animal cell like we learned back in chapter 3, but it does have some of those same features. So if we take a look up here at this big part, this is called the cell body. Kind of this, I'll box it in right here. So all of this is a cell body. If we just looked at that part, it would look kind of like a regular cell. We have a nucleus, we have mitochondria, we have this, what's called Nissl substance is the same thing as what we learned as RER, rough endoplasmic reticulum. It's got a cytoskeleton, so it has all these things. They might have slightly different names, but it's the same idea as a eukaryotic cell. But once it starts, if we take a look outside of that box, here we are going to see some things that are unique to axons. So we have this long extension coming off of it, and this is called an axon. So here's the axon right there. So an axon is where this nerve impulse is going to travel. It's generated in the cell body, and then it sends this signal very long distances down the axon to then communicate to whatever the neuron is attached to. So the very, very important part of a neuron, of where that initiation starts, is called the axon hillock. It means little hill. So I guess it kind of looks like the cell body is a little hill down to this little skinny axon. So when we take a look at the physiology of a nerve impulse, this is going to come into play a little bit more. So right now we're just learning the anatomy. So we have the anatomy of the cell body, we have the axon hillock leading into the axon, which will then go into what we call axon terminals. It is the axon terminals that are going to communicate to the next cell, which could be another neuron, it could be a muscle, it could be a gland, those effectors, right? Or just another neuron in a circuit. Okay, so this is the major structure of a neuron. The one thing that is different besides the axon, these other little kind of stringy things sticking off the cell body, these are called dendrites, these branches. The word dendrite kind of means like tree or tree branches, so that kind of makes sense. So it is the dendrites where receiving information is going to come in. We'll use our word afferent, right? So incoming information is picked up by the dendrites, and then that signal is going to be leaving the neuron, and we could use this term efferent. Okay? Afferent is bringing in, efferent is sending out. Right? So I have some pictures, just clean up this diagram a little bit, some micrographs on the right-hand side to illustrate some of these things. So the top picture kind of looks like something you would see in a sci-fi movie, but it really is, I'll use a yellow. So here's the cell body, here's all the dendrites, and then here's the long axon branching out that way. So it's really kind of spindly. The other picture that we see here is a cross section of the axon. So let's say I was going to take a section right through that part of the axon, through that Schwann cell. Remember I mentioned Schwann cell in the last video. It is the cell that does the myelination on these peripheral neurons. So inside, so over in this micrograph, looks like green and yellow. So I'm circling the axon right there, and then I'll use blue right there. So all these green layers, it looks like almost like an old record. So all of this thick layering, and here's a bigger one here, this is the myelination. So you can look really closely and you can see it's layer upon layer upon layer, and that's what creates the myelination in the peripheral nervous system. So it's the same with those oligodendrocytes in the CNS. It's the same function, just different cells. They wrap around, and you're going to get that myelination. Okay, so that's the structure of a multipolar neuron. Has the dendrites bringing information into the cell body. The signal gets triggered at the axon hillock, sends its signal down the axon towards the axon terminal to deliver that message to the next cell. Okay, so now this is taking a look at a functional classification. So here we're going to use those afferent and efferent words again. So if we are picking up information, so information is coming in here, and we want to take it towards the central nervous system, we are going to be an afferent neuron. Remember, afferent is like sensory, bringing information in. So this blue neuron is considered an afferent or sensory neuron. Okay. Now, if we have our dendrites here on this red neuron, and we're sending information out this way towards the periphery, towards a muscle or a gland, this is your efferent transmission, and it makes this a motor neuron. So we have sensory neurons bringing information in, we have motor neurons sending information out, and then right in the middle, within the central nervous system, we have what are called association or interneurons. They're just processing. They're neither afferent or efferent. They're kind of in between, almost like a relay race, right? So we just got done watching the Olympics. So you have one runner bringing their information from the body into the central nervous system. This runner is going to transmit that information to the next neuron, which will send it out to your periphery. So these are functional classifications. There's three options here: afferent for sensory, efferent for motor, and association neurons for the kind of connectors, the in-betweens. Now, if we take a look at structural classification, we've already identified the multipolar neuron, right? So we saw the cell body with all of these dendrites sticking off and one long axon. That's why it's called multipolar, because it has multiple little poles or things sticking off of the cell body. The next class is called a bipolar neuron. So it's got the kind of bulgy cell body in the middle, so that's going to have the nucleus and the mitochondria and stuff. And it has two processes coming off. One half acts as the receiving part, and the other half acts as the sending out part. Where on a multipolar neuron, it's all kind of, the receiving part's all squished around the cell body. In a bipolar neuron, it's more evenly distributed. Where multipolar neurons are very common, bipolar neurons are not very common. They're pretty rare. They're only found in special senses like your vision and your olfaction. And then lastly, we have what's called a unipolar neuron. Okay? So if we got to look close, but we have the cell body and then one short little process sticking off of that cell body. That's the uni. Uni means one. So this unipolar little process coming off, and then that's going to split into this long axon. The dendrites are just right here. So there's dendrites without a cell body. And then we have this whole axon sending the information towards the destination, a muscle or a gland or another neuron. So those are structural classifications. So we're most likely going to be seeing multipolar neurons for the rest of this discussion, just because they're the most common in the brain and the peripheral nervous system. But just know that there are two other appearances of neurons, the bipolar and the unipolar. This diagram or this photo micrograph should look familiar. That's what you used when you were learning about neural tissue back in histology. And then this is a drawing of a very dendrite heavy multipolar neuron. So the cell body is right here. All of these are dendrites receiving a lot of information, and here's the single axon coming off. So this is considered our multipolar neuron. Or maybe that's bipolar. That's actually hard to tell because it sure looks like there's only two processes coming off. Either way, the dendrites are numerous. So that means this particular neuron, whichever type it is receiving a lot of information before it sends it out on that skinny little axon. Okay, so now we're going to get into this process of nerve impulses. Sometimes they're called action potentials. You might come across that word. They're the same thing. These electrical signals that are running down the length of the axon. To understand what's happening, we have to kind of set the stage. So this picture A is showing you the resting membrane. And so if you remember, we had cells. Let me see if I can get my pen working here. So in a basic cell, we had like a nucleus, and we had the outside of the cell, and we had the inside of the cell. And if you remember, our phospholipid bilayer, right, our plasma membrane was impermeable to polar things like ions. So if you were going to move something in and out of the cell that had a charge on it, you had to have special channels, like You had to have these facilitated diffusion, these channels that would open and close at particular times. So that's what we see here in the membrane of an axon. Axons are excitable, very much like those muscle sarcolemma that we saw back in chapter 6. They have the ability to carry out these action potentials, these nerve impulses along the surface of the membrane. So in a resting cell, what we see is the inside of the cell is more negative and the outside of the cell is more positive, right? So this is my cell. I have more negatively charged ions on the inside and more positively charged ions on the outside. Now, looking at the ions that they have listed, it might be a little bit confusing because they're both positively charged. We have sodium plus on the outside and potassium plus on the inside. That just tells you there's going to be an exchange of those two ions, not that the potassium is leading to that negative charge. There's other things inside that are making that overall negative charge. It's not the potassium, but the potassium does play a role in returning the membrane back to rest. So you get a stimulus. So let's say the cell body, some information is being picked up by our dendrites. It's stimulating the cell. And right here, remember that axon hillock, that region right at the very beginning of the axon? That stimulus is going to show up. And what that stimulus does is it opens special channels that allows sodium to rush in. Now, remember, sodium likes to go from, anything likes to go from a high to a low concentration. So when we have a lot more sodium on the outside, whenever that channel opens, because the stimulus showed up, the sodium rushes in. And it rushes in so much. Take a look at what's happening in this picture. So much sodium rushes in that we actually flip-flop that orientation of the cell, where the inside now becomes more positive and the outside becomes more negative. That in itself is the signal. This flip-flopping of these charges on the membrane creates this signal that can then move and propagate down the line. So then as that sodium is rushing in, it will continue to rush in and continue to flip-flop those charges and allowing that signal to travel down the axon towards the axon terminal. Now, how do we get back to rest? We can't just keep the neuron be flip-flop charged the whole time. So here's where the potassium comes in. So now, because there's more potassium on the inside than the outside, a different set of channels open, allowing potassium to leave the axon. So what that does is so much positively charged potassium leaves that we go back to our original state, which is negative on the inside, positive on the outside. This process of going back to normal is called repolarization. The process of flip-flopping is called depolarization. Okay? So we start being polarized. Polarized means opposite, right? So the negative and the positive, they're polar, opposites, okay? When the signal comes and the sodium ions flood in, we depolarize, so we flip. And then when the message is, heading down the membrane and we're going back to rest and the potassium leaks out or floods out, we repolarize. We go back to our resting state. But in the meantime, that signal continues to travel all the way down the length of the axon until it reaches the axon terminals to communicate to the next cell. So this is the basics behind a nerve impulse, or what we call an action potential. And it's all about changing the charge. So we have to start with more negative on the inside. The signal shows up, sodium rushes in, we become more positive. That signal continues down the axon, and then leading up behind it, potassium leaves, flips back to the more negative on the inside, and then we're back to rest, allowing another action potential to move down that axon if it gets stimulated. So this graph is showing you kind of the same idea, but kind of mapping it out, showing you those flip-flopping of charges. So let's say, we said the inside of the cell, right? So if here's our axon, if the inside was more negative and the outside was more positive, right? So that was our resting state. We actually are resting at a value that's called -70. So that means the inside is 70 units more negative than the outside. It's all relative. It's all comparing the inside to the outside. Now, if you're just in a resting neuron, I'll pick a different color. So I don't want to do red on red. So if you're just hanging out at resting potential, you're just staying at negative 70 millivolts. But then remember when I said the signal came in, this little stimulus, that's this black arrow right here. If that stimulus comes in and triggers sodium to come into the cell, that's going to cause more positive charges to come into the cell, right? So we're on that way to flip-flopping. And there's a very special number here called threshold. It's that magic number that's going to allow for all of those rest of those sodium gates to open. So if we reach threshold, now we're going to rush in all of those sodiums and take a look at what happens. We go all the way to a very positive value. So that's going to turn these positive, right? So positive, positive, positive. Get in here and make all my negatives. So then the outside is going to be more negative, okay, in relationship to the inside. So then our sodium is done rushing in, and now we're going to get our potassium. Remember, potassium likes to rush out, kind of following behind this action potential. So now take a look at what happens when all of that potassium rushes out. We go back to being negative on the inside, right? So I'm now going to erase all my positives because we're no longer positive. We are back to being negative on the inside and positive on the outside. When that potassium leaves, that's called repolarization. And then there's a period of time here called the refractory period. It's going to take a little bit of time. We actually overshoot our resting potential and get all the way down to negative 90 in most cells. But then we have our active transporter sodium potassium pump, pumping the sodium back out, pumping the potassium back in to get everybody back to rest. And eventually we will get back to rest. Look at my positives up here, my negatives in here, and we're going to be back to resting state. So this is a graphical representation of this nerve impulse, and the earlier picture was more of like what's actually happening on the neuron. So between those two diagrams and reading through the book and watching this video and maybe finding some helpful videos on YouTube, you should have a good grasp of how to explain or describe or diagram, draw your own picture of these nerve impulses. Okay. So then what happens once that nerve impulse reaches the end, the synapse? What do we do? Well, here we have a picture of a neuron communicating to another neuron right over here. Right? So we have the signaling neuron and we have the receiving neuron. So we're going to take a super duper close-up look at that connection between the cell axon terminal and the receiving cell. And this should look really familiar to you. It looks almost just like a neuromuscular junction, and it functions exactly the same way. So this should be somewhat review, but instead of going from a neuron to a muscle, we're just going from a neuron to a neuron. So we have our synaptic vesicles, okay? So the synaptic vesicles, let me pause real quick. Sorry, my computer was lagging for a bit. So we have our synaptic vesicles filled with their neurotransmitter. It could be acetylcholine. There's a whole bunch of different neurotransmitters. We can stay with ACH if you're comfortable with that. When this signal comes down, so here's this action potential. It triggers that exocytosis. The neurotransmitter floods the cleft, binds to those sodium channels, opens the sodium channels, and sodium floods in. That's the stimulus that's going to trigger this next cell to do its action potential. So it's just, like I said, it's this relay race. We're going from an electrical impulse along the axon to a chemical messenger, say ACH, across the synapse, because remember, they don't touch. And then once you have all that sodium flooding in, you're going to trigger another action potential because you're going to flip-flop those charges and be back to that last topic we just got talking about. So this is what we call synaptic activity or communication at the synapse. This picture here is an electron micrograph of a synapse. So this cell here is the axon terminal, and you can see all the little circles. Those are the synaptic vesicles, and they're just jam-packed full of acetylcholine. And then this cell here is the receiving cell. So that could be another neuron. It could be a muscle. I don't know. And then that space, this tiny little space, this is what we call the cleft. Okay. So those synaptic vesicles, once they fuse, like right here, they'll be releasing their neurotransmitter into that tiny little space. All right, so that's our synaptic activity. All right, so the next topic, as we kind of look a little bit zoom out in our neuron world, we're going to take a look at reflexes. So you may have heard of reflexes before, have had your reflexes tested, like at the doctor's office when they whack your knee. That's reflex action. So there are 5 major components to what we call a reflex arc. So we have the receptor. So this is what is picking up the signal. We have the sensory neuron, which is bringing that information towards the CNS. We have the integration center, which in this case is the spinal cord in this picture, but it could be the brain. We have the motor neuron, which is step 4, so that's sending the information out. And then lastly, we have the effector. So in this case, it's illustrating like a pinprick, right, on your finger, some kind of pain stimuli. And typically what happens when you feel pain is your body pulls away. The special thing about reflexes are they do not need any processing for them to work. They don't need any conscious processing, I should say. There's local processing, but no thought. You don't have to, like if you're feeling pain, you don't have to think about, oh man, that hurts. I'm going to send a signal to remove my hand from the hot pan. It does it automatically before you might even feel the pain stimuli. And then you're like, oh, I just burnt my hand, but your hand's already been pulled away from the damage. So reflexes are very protective of your body. They can allow you to move body parts away from painful or damaging stimuli without needing to process it through your conscious part of your brain. Okay? So just to note, this effector, in this diagram, they're showing a muscle, and those are the reflexes that we are most common with or familiar with, is like pulling away from painful stimuli like a hot pan or you're stepping on a tack. But please know that not all reflexes are just muscular. There are also hormonal reflexes and there are glandular reflexes. Think about when you're, anticipating food. You think of your like food is coming and your mouth starts to water. That's a reflex. And the effector is your salivary glands. Your stomach does the same thing. When you're thinking about food, you have this almost reflective action. You don't have to process and make the decision to secrete these things. It's happening reflexively, not reflectively. That's like shiny. Okay, so they can include muscle, cardiac, skeletal muscle, smooth muscle, cardiac muscle, and glands, all of those effectors. We have different kinds of reflexes. So the ones that use our skeletal muscle are called somatic reflexes, right? Like the knee-jerk reaction. But we also have autonomic ones that will use those uncontrollable, I don't want to say uncontrollable, those involuntary effectors. So then this picture down here is just showing the same thing, just a slightly different visual. So here's your sensory receptor. Here's your sensory neuron. Here's your integration center. Here's your motor neuron, and here's your effector. So being able to recognize these things in different types of diagrams is going to be helping you learn this path of the reflex arc. Okay. So here is the last slide we're going to take a look at, is showing you 2 different types of reflexes, how they're classified. This first one on this side is called a monosynaptic. And mono means one, and synaptic means synapse. So if we take a look, the sensory neuron synapses directly onto the motor neuron right here. And then the motor command goes out. So this is what's happening in your knee-jerk reaction. This one over here is called polysynaptic, because poly means many or several, and synaptic means synapse. So if we take a look at the integration here, integration center on that side, there's at least 2 synapses. Now these are pretty simplified. You can have some polysynaptic reflexes that have hundreds of synapses between them. But if you have at least 2 or more synapses in a reflex arc, you have a polysynaptic reflex. So this is going to just bring a little bit more complexity to the circuit, but it's still automatic. It's going to happen without conscious processing and thinking about it. In this case, it's pulling away from the pain. This is called a withdrawal reflex. So withdrawal reflexes are a little bit more complex because of that interneuron that gives it that polysynaptic classification. Okay. All right, so that wraps up our neuron video. I think we're going on into, let's see, the central nervous system. So we get to talk about the brain next. All right, see you next time.